Our next speaker is one of my colleagues, Peter Kasson from the University of Virginia. I've known Peter for a number of years, and he's going to talk with us about better inference through chemistry, knowledge-guided inference of biomolecular kinetics. And so, Peter, if you can pull up your slides.
Great, thank you. It's a pleasure to participate in this workshop. So what I want to talk about is a set of approaches to understanding the kinetics of macromolecular machines using sparse experimental data and two different types of sparse experimental data. One is where we have structurally specific data. So on, for instance, proteins, if we have labels at sites we know on those proteins, and we have then information about how those labels interact with each other and give us distance information and distance constraints and how they evolve over time. And that yields one set of methods to try to understand how macromolecules have structural and kinetic diversity. The other is a class of more challenging data that I would say are structurally indirect, where we have reporters on a biological process. And the example that I'll use, and we'll go into more detail, is for looking at viral entry, where we can measure this on a single virus level using fluorescence dequenching. And so then we get fluorescence signals that reflect a state change in the biological process, but are not trivially assignable to specific molecular states. And we're working on actively developing methods for kinetic refinement of these using statistical learning, also using some physics-based methods. And so there's, for both of these, there's a lot of interplay between the sort of philosophically related areas of statistical learning and statistical physics. What I want to share with is our current state, which has maybe somewhat more mature methods for statistical physics-based inference than statistical learning-based inference. But we believe very strongly that these two work together. And I also want to explain some of the challenging nature of the problems. So why we care about this is to try to infer kinetics and mechanism across scales. And so what you see on the left is a movie of the kinds of experiments we do on single virus entry. These spots are each individual influenza viral particles. And as they, in the movie I played, as they got brighter, those were the particles fusing and becoming infection competent to insert their genome into a cell. And at a much more detailed level, we have information from, say, cryo-electron microscopy on the upper right that gives us the architecture of these viral particles, and then very detailed molecular models, as are shown on the lower right, that are models that can integrate some structural data, but nonetheless have a very substantial gap in spatial and temporal scales between these and the sorts of inference of kinetic models we can do based on molecular dynamics trajectories and the sorts of inference we can do on kinetic models based on viral fusion trajectories. Why we care about this is a combination of mechanistic insight and therapeutic design, that we have data showing that antibodies to influenza and other viruses can bind to essentially the same place on the viral protein, yet work differently. And this is perhaps not surprising because these viral spike glycoproteins that I think you'll also hear a lot about in the next talk are proteins that act by taking on a number of different shapes and by mediating multiple steps of attachment and entry of the virus, and therefore looking at it in a purely static model to try to infer mechanism and also try to optimize therapy ends up being likely insufficient. And so the first problem that I want to talk about in this regard is a problem on the way to kinetics, and I'll explain a little bit where we want to go, but of structural ensembles that both for viral glycoproteins and for many other flexible proteins in biology, that we can sometimes resolve one or a few states at high resolution with structural information, but the full ensemble of states and often the intermediates contain much of the important mechanistic information for figuring out how these function and also how to engineer them. And however, for getting at structural ensembles, we often run into a problem of sparse experimental data, that the experimental techniques that report on structural ensembles tend to yield a small number of degrees of freedom relative to the number of constraints required to resolve molecular structure, yet they can give distributional quantities rather than just mean values for these degrees of freedom. And so that's shown on the left with some single molecule FRET data from the laboratory of my colleague Walter Mathis. What we want to do is be able to take these sparse data and use them to infer the structural ensembles of, or infer as much information as we can about the structural ensembles of the molecules that they're reporting on. And one of the goals we have is transferability, that if we have extensive information about a molecule in one environment or under one set of conditions, can we use sparse and potentially expensive to acquire data that are many fewer data to then transfer our knowledge about the structural ensemble in one environment to another environment. And from statistical physics, if our if our methods for adapting our structural models from one environment to another satisfy maximum entropy properties, then that is then the least perturbing transformation of the model from one to another, and thus represents the best we can do to understand the protein in its new environment. So that's some of the motivation. One of the methods that we developed, and again, this is more physics-based inference, is to be able to estimate these heterogeneous conformational states based on sparse distributional data. One of the challenges is just refining this in a stable manner when the data are relatively heterogeneous, that many of the prior approaches work well when you have probability distributions that are continuous or near Gaussian. And so we developed an approach that is very similar to stochastic image formation, that essentially takes an initial model of the structural ensemble of the protein. This model can be a bad model, and then draws random samples from that and random samples from all our experimental data distributions, and essentially refines each sampled conformation against a distribution that looks like a delta function that's a random sample from the target from each of the target distributions, and then collects all of those and uses that ensemble to update our estimate of the structural ensemble of the protein of interest. And we can do this fairly well, and indeed outperform for these challenging distributions prior state-of-the-art approaches. And so this then gives us the tool to do what it says are the interesting pieces. And one is, again, utilizing some properties of statistical physics. If the experimental data are acquired separately in different experiments, they then contain no information about the correlation between them. And most approaches in the field have treated those then as independent, and the joint distribution as simply the convolution of the individual distributions. Whereas we know that in many cases, if you're measuring a protein that's flexible and moving in different ways, that there has to be correlation between the different measurements. And one can illustrate this with a very simple toy model of an alternating transporter shown on the left that can take two conformations and the sort of intermediates between those, but where the two measurements are very clearly coupled, and where if one just assumes that you get the two individual distributions are the joint distribution is the convolution of the two individual distributions, you get something very different from the true analytical distribution shown in panel C here. What we can do is to essentially perform physics-informed inference, that estimating the non-equilibrium work required to restrain this, the system to any point in this joint distribution yields the probability of that point in the joint distribution when averaged over many trials. And so again, via sort of a stochastic approach, we can build up the correct joint distribution after a large number of runs. And that's shown here. And then we can apply this to a flexible protein such as syntaxin, shown on the right, which is a neuronal protein involved in neurotransmission, and yield elements of the structural joint distribution, both states that would be, if we don't know any of this correlation information between the experiments, we come up with some states for the protein that are actually physically disallowed, and certain other states in this joint distribution are much, much more likely. And so we can greatly improve these estimates. What we really want to do with this is to have a statistical inference cycle for refining structural ensembles of flexible proteins, where we know how to best incorporate experimental data and then can best choose the experiments, given that these experiments are relatively low throughput and relatively expensive to perform. And so the next component, I've told you about some refinement approaches, is to ask, can we identify the most informative experiments to perform to further refine conformational ensembles? And so for this, we took a relatively, in the information theory community, a relatively well-identified quantity, which is a maximum relevance, minimal redundancy quantity between different potential labeling sites on a protein. And we showed that even given very noisy starting estimates for conformational models, we could select experiments, use the resulting data to refine those conformational ensembles, and substantially outperform experimental selection by a trained spectroscopist, in large part because our estimates both could eliminate redundancy better, and we could also know when to take certain risks in experiment selection much better than a spectroscopist who ended up being much more conservative in those cases. And so then this gives us a way to complete that cycle and have methods for inferring the probability distribution of structural ensembles given experimental data, and methods for selecting experiments to go on to further refine these structural models. The next cases, of course, will be to use many of these experiments, but not all, also yield dynamic information. And that's going to require a little bit more statistical inference. That's something we're working on very actively right now. I'd also say that, you know, I've shown some non-viral examples for this. We're also working on HIV glycoprotein and other viral glycoprotein structural ensembles, and can match experimental data very well with that. The challenge for these larger scale and non-structurally specific experiments is that the states are highly degenerate, that these fluorescence dequenching data that I've shown you, if we look at a single virus trace on the upper right, it's essentially a step function. And so we can measure a state change from not entry competent to entry competent, and we can build this up into a cumulative distribution function over many trials. But there are many molecular states and indeed many, often many kinetically important states that all have this initial emission value as opposed to the final emission value. And this degeneracy makes it very difficult, as I'll sort of briefly show you in the final few minutes, to train a lot of traditional statistical inference models. What we've done in the past is traditional sort of fits where one can postulate a kinetic model that corresponds to a series of differential equations and perform inference-based fits of these models. But this is really very, very driven by specific hypotheses. This is for Zika virus, and can indeed yield substantial insight from this. But we'd like something more general. And also, furthermore, for Zika, we can change a parameter and see differences in a number of rates, but the functional form, this number of intermediates in the existence of an off-pathway state, which we'd like to exploit for inhibition of viral entry, is all the same. However, when we look at influenza, if we change some of these parameters, as you see on the left, the functional form actually changes, and we can quantify this using methods that were developed for single molecule enzymology. And we can see this change both when we change temperature and when we change other physical parameters in the experiment. And what we'd really like is an inference framework to be able to extract the dependence on these physical parameters and encode what we know about how model families should depend on these parameters in a principled manner and in as general a manner as possible. This is challenging. I sort of show you the success here. This is very much work in progress. That hidden Markov models with a number of constraints can fit some of these very well, but others, because of the state degeneracy, it becomes very difficult to converge traditional models, and a number of the sort of encoding prior knowledge and regularization approaches become very important. What we're working on is one step beyond that, to be able to properly encode shared parameters across the model families. And what differs from sort of off-the-shelf models here is that the time series update step reflects the chemistry, that we know how update steps should reflect, say, temperature or some of these other things. And so it's not just a linear dependence. There are nonlinear dependencies in the update step that make life a little more complicated, but very interesting. So I'll wrap up there. I'd say we're actively using, particularly where there's sparse data, a combination of statistical physics and statistical inference. State degeneracy for these sorts of problems is extremely challenging. And the thing that I find most exciting is being able to encode chemical knowledge within an inference framework. Pardon me. With that, I'll just thank the members of my team, our funders, and all our collaborators. And if there's any time, take questions.
Excellent. Thank you very much, Peter. I'm going to take session organizer's prerogative, and I'm going to ask you a question. You mentioned multiple times the challenge of degenerate states and sparse data. The question that was on my mind is how much of this is just the essence of protein dynamics that you are studying, and how much of it are a function of the probes that you're using to assess those states? In other words, if you had infinite knowledge about everything, with atomic resolution, what the protein is doing, would that resolve the sparsity issue? Or simply are the biophysics of these proteins such that they're just these rare occupancies that you'd never catch them no matter how good your detection reagents are?
If one had the ability to, measure everything in high time resolution, we should be able to resolve these. I think the challenge is that the nature of the measurements, and even some of these measurements are perturbing by themselves, is that they can't resolve particularly transient states. And then also some things are degenerate to the accessible probes.
Does that open the door then that better probes or better measurement technologies may eventually resolve that challenge? Or have you hit a type of measurement limit for where some of those are? In other words, how fast you can snap the camera, or how many times you can look at a fluorophore before it starts bleaching?
I think there will be some fundamental limits. I think what we can do is also increase our ability, particularly since for many of these we actually control the experiment design as well, increase our ability to do experiment selection. And so if we can use the models and the uncertainty in the models to guide probe design and experiment design, we can take this farther. There will be some fundamental limits, but we're not quite there.
Okay. All right. Thank you again, Peter, for a nice talk.